Hi guys. Pretty sure the last time I did this I abruptly cut it off because the janitor came in and I feel silly sitting here talking to myself. So thankfully I had just finished, I hope. But I just had to cut it short without saying goodbye. But we are getting closer to finishing these notes, kind of. But we ended with talking about transpiration and the evaporation of water from plants and how that moves. So if you've never seen something like this, it's kind of cool because you just stick the plant in this tube and there's water in here, and so water will leave the leaves, and then you can see how much water was evaporated by watching the water go down this calibrated pipette. So that's how that works. The next thing you'll need to know about is how sugar actually moves through the plant. We've already talked about how water moves through the plant, but we haven't really talked about how sugar moves through the plant. Water moves from the roots up, but sugar actually has to go from the leaves down the plant. So this is all described in what they call the pressure flow model, or source sink model. So sugars are going to move from where they're made down to the roots. So they just call that source sink. I usually don't hear the pressure flow model too much, but it just goes in waves. I won't hear about it for a couple years. They'll use the source sink words, and then all of a sudden they'll go back to pressure flow. So just be aware that it's the same thing. I'm going to try and mess with the focus on here. There we go. So the first thing that has to happen is sugar has to be made. So here we have a leaf cell, and that leaf is making sugar. It doesn't have to be sucrose, obviously, you guys know that, but that sugar will actively move into the phloem. So even though there's already a high concentration over here, it's going to keep pushing it into the phloem. Remember, xylem transports water, which is why you just see the water right here going up. And minerals, by the way. But water flows passively by osmosis, and it's going to create an area of high pressure at the source. So water's moving this way, and it's because of osmosis, it's going to travel over to the phloem, and that pressure will push the sugar down. So really it's because of water that the sugar moves in a downward direction. Just moving from high to low pressure. So the highest pressure is going to be, sometimes they ask, like, identify where the most pressure is. So of course the highest pressure, whoa, the highest pressure is going to be up here, and low pressure down here at the sink. So if we were to summarize that, in a very simplified way, short, concise. Leaf produces sugar. Sugar goes into the phloem. Water will then go into the phloem. And all that pressure from the water will push the sugar down. So we've spent 5 minutes talking about this, but it's really pretty easy. We know that leaves make sugar through photosynthesis. The sugar will move into the phloem actively. Water will then move into the phloem, and then that water pressure pushes the sugar down. They will also ask you how minerals are going to get into the plant. So if you look at this picture, they have clay here. That's the same thing. They're talking about soil. And a pump is used by the cell to move hydrogen ions out of the root cell, and that will set up an electrochemical gradient. So in this picture, always get yourself oriented. Outside, inside. Makes sense that the soil is on the outside. And so what happens, there's a pump right here. We know it's a pump because it's using energy. We're using ATP right here, and we're moving those hydrogens out. And the second we do that, things can now move in. So potassium is one of those things. You can see the potassium in the soil. Another is calcium. And both of those are going to flow down the electrochemical gradient into the root. Some things can also be co-transported with hydrogen as it passively moves. So we just pumped a ton of hydrogen out here. And so some of them are just going to naturally, passively, because there's a higher pressure here, just move back in. And then there are some molecules that are attracted to that hydrogen and will move back in with it. And chlorine is one of them. So if we were to kind of organize this, this is letter A. The pump is used to set up the gradient, moving those hydrogens out to where the soil is. Then positive ions, like potassium, are just going to go down the chemical gradient into the roots, and some are actually attracted to the hydrogen and are co-transported with it. So those are the 3 steps. I don't know why I waited so long. It probably took you 2 seconds to write letters. Felt like I needed to wait a decade. This next section is all about plant communication and hormones. And so some of this we've already talked about, how hormones work, and it's the same for plants as it is for animals. So you'll still see that signal transduction pathway. So some of this hopefully sounds just familiar to you and what we've already talked about. So anytime we talk about tropism, that's just response to something. Man, I really hope I don't say a million times in these. It feels so weird to just sit here and talk to myself, and a lot of times it feels comfortable to fill the silence with Anyway, if we have a response, there are several different types of responses. If it's gravitropism, response to gravity. Phototropism, response to light. Thigmotropism, response to touch. So they're going to talk specifically about light and how it affects the plant. There is a hormone specifically involved in plant growth called auxin. The details of this are on the next page, but auxin works like any other hormone through the signal transduction pathway that you guys all know about. So we could imagine this as our auxin because it's the hormone. Remember, the outside molecule is also the primary messenger. And then there's going to be some sort of molecule in here that's activated. That's the secondary messenger. And in order to get a response, you have to activate transcription factors. Just as a quick review, remember transcription, DNA to RNA. So those details are specified down here. So we have a receptor right here, usually a G protein linked one in the cell membrane, and that's going to bind to that hormone. That causes it to change shape and activate another molecule inside the cell, which is the second messenger. And if you remember, there was an example of a second messenger that was really common in a lot of cells, and that's cyclic AMP. And that's just going to keep on activating other molecules by phosphorylating them, eventually leading to activated transcription factors. And turning, that means you're turning on genes. So anytime you summarize this, you might want to mention both factors. The fact that you're activating transcription factors, which ultimately means you're turning on a gene. Remember this picture where you have the kinases that are activating each molecule in this transduction pathway. Adding the phosphate to activate them. Take the phosphate off, deactivate. Okay, so phototropism is actually a great example of how secondary messenger system works, because it's going to use auxin and then use other molecules to create growth in our plant. So the very first thing that has to happen is light shines on the plant, and that causes auxin to migrate to actually the shaded area of the plant. Via diffusion. At the cellular level, auxin is binding to the receptor on the membrane. That's going to activate secondary messengers inside the cell. The first messenger activates a proton pump, and what that does is it creates an acidic condition that loosens the cell wall. And then another messenger activates the Golgi to send out vesicles that will help with the growing and elongation, or elongating the cell wall. Another one activates DNA binding protein that will activate transcription factors. Not sure why you're not given more space for that, but activates a transcription factor that will lead to the production of growth factors. So let's kind of review those steps again. This is a zoomed in picture of what's going on here. All that's happening is your auxin is binding. That was step A up here. Well, actually, that looks like B. Let's say that's B. So auxin is binding, and that's going to activate several things to happen. So there's a proton pump right here that will loosen the wall. So that's letter D. If you want to keep going to, some of you guys aren't very visual, you just read and learn. Some of you guys really like pictures and seeing what's going on. So that was D, the loosening of the wall because of the pump. This is the Golgi, and it's releasing molecules that will be used to build that growing wall. So that's E. And then another one, another messenger over here, goes this way, and it activates transcription factors that will produce growth factors. So all these steps are described right down here. So what's happening right here is kind of interesting, and it's hard for me to show you what's going on with a video. But what's happening is the auxin causes cells on the shady side to elongate faster than those on the lighter side, and so that causes the stem to curve towards the direction of the light. So if I have a plant, I don't know how to draw this. I'm just gonna make it a stem. So if light is shining over here, then the auxin will cause the cells on the shady side to elongate faster. So if you imagine these cells all along here are going to elongate and push the plant this way, and there's going to be growth like this. But as the sun moves, it's going to do the opposite. So the sun will go this way, and then it'll start to elongate on this side and not on that side. So on the shaded area, the side that's not hitting the sunlight directly is going to get longer, and that pushes it in the opposite direction. Hopefully that makes a little bit of sense. I think I usually have a good video that explains that better, but hopefully I can find that and include that somewhere in these lesson plans. Negative and positive feedback are just a very common theme in AP biology, and this is an example of negative feedback for plants. And if you guys remember, I don't remember if we talked about negative feedback already. I know we've mentioned positive, but negative feedback is when the result of a process will shut off the initial process entirely. So in this case, auxin migration to the dark side will cause elongation of that side. But then the plant is going to, that causes the plant to bend to the light. Sometimes this video pauses and I don't know if it's still recording or if it actually pauses and isn't recording me. So if I repeat myself, it's because I don't know if it's still recording me or not. But the elongation causes the plant to bend to light, but then light hits the dark side, and then that will keep the process going just on the opposite side. Okay, and then the last thing, hormones are going to also be involved with cell death, and one major hormone is ethylene. The specific way it does that is because it promotes breakdown of cellulose and starch in the plant tissue. And as that happens, so this is an example of positive feedback. Because what happens is ethylene causes breakdown of the tissues. When those cells break down, that actually releases even more ethylene. So more ethylene will create more breakdown, which creates more ethylene, which will create more breakdown. So that is an example of positive feedback. So you're going to amplify a response until your final, I mean, they always give positive feedbacks, but as an example of one thing that reinforces our initial impact, but you know, of course you have to end it at some point. So when the final result is achieved, then it can stop. If you were to be given this picture, they might ask you something about what leads to the cell death. And it might say something like, what is something that, what does ethylene inhibit? And if you guys remember, when you first see a picture like this, it might be a little intimidating, but it's always an easy answer. So it'll say something like, what does ethylene inhibit? And you can look at this stop line here, and it's going to inhibit MT2B, whatever MT2B is. So anytime you see a direct arrow, that means activates or promotes. Anytime you see the stop line, it's inhibit or stop. I hope I didn't just say that backwards. So yeah, just as a final note, as I'm doing this, really feel free to email me if you guys have any questions. And this is probably more like college than any other part of the year, because you are in charge of your own learning, and if you don't understand something, look it up a little bit more. If you don't understand the auxin, look it up. There's plenty of stuff online, and even though I have a baby at home, I'm still available. So please, please, please email me if you have any questions. So I hope you guys are doing well, and I miss you guys.